So first of all, we will write the topic neural control and coordination. I'm using white background to ensure that you can get the PDF and get a printed copy if you want later on. I'm zooming and writing so as to ensure good handwriting. So neural control and coordination, lecture 1. First we will understand about the neuron. What is the neuron? How does it look like? So basically a neuron is divided into two sections. The first section is the cell body. The cell body, which is the cyton, which is called the cyton, and the cellular processes which are further divided into the axon and the dendrites. Axon is long and cylindrical. While the dendrites are small and branched. So these are the basic, this constitute the neuron. There are different types of neuron as well, but let us first understand the structure of the neuron. We will draw the axon first. Yeah. So okay, we've drawn the structure of the axon. Now I will draw the cell body. I will explain to you the everything, but first let me draw the outline. Okay. Now this is the cell body and the cyton. So I will just show you using a different color. Let's use red color in this white background so it will look good. So this part actually is called the cyton. This part is the cyton. This cell body is the cyton. And if it is a cell body, then definitely it has a nucleus. A cell has a nucleus where the genetic material is stored. We all know that. And from the cell body, these are the projections, cellular projections, the cellular processes, which are called the dendrites. These are called the dendrites. I am showing 3 so as to ensure that there is proper understanding of the structure and it ensures clarity. And now I am drawing the end part. These structures, this one, these are called the synaptic end bulb or the synaptic knob. This one is the axon. Now let's draw the synaptic vesicle inside the synaptic knob. So the synaptic vesicle, the synaptic vesicle is the storehouse of the neurotransmitter. Now inside the cell body or the cyton, we will draw this. These are called the nasal granules. Let me draw the outline first. It will be easier for you to understand then. I'm just telling you, for those people who have already studied they will understand everything. While for those people who are seeing it for the first time, they just wait for a couple of minutes. Over the axon, you have the myelin sheath. It is an insulating layer which doesn't allow passage of ions. So this is the myelin sheath. Lots of noise, So this is the myelin sheath. It will look good actually. And over the myelin sheath, we will show the Schwann cells. Obviously they're cells, so they have nucleus. And over this entire structure we have the neurolemma. Okay, let's correct this part. So okay, I've drawn the basic structure of neuron actually. Now let's go into the leveling and understanding part. So this is the basic structure of the neuron, and this is the cell body, the cyton. These are the dendrites. Small cellular projections. And this is the nucleus. Obviously I'm showing you, I'm not writing it so as to ensure clarity. This is the nucleus obviously, and these are the nucleus of the Schwann cells. These are the nuclei of the Schwann cells. These ones are the nuclei of the Schwann cells, and this part is called the axon hillock. The axon hillock is the most sensitive part of the neuron. The most sensitive part of the neuron. Now, this part is called the axon terminal. Axon terminal. These are the synaptic knobs. Synaptic knob. Synaptic knob is also known as synaptic end bulb. And inside the synaptic end bulb are the synaptic vesicles. The synaptic vesicle is the storehouse of the chemical neurotransmitter. For example, if we have acetylcholine will be stored inside the synaptic vesicle. We will understand later on how the synaptic vesicle fuses after the influx of the calcium ions due to the impulse that is being carried on, and the depolarization and repolarization are occurring. And after fusing due to exocytosis, that neurotransmitter flows into the synaptic cleft and then gradually into the new neuron, and that's how the impulse is passed on. This cylindrical part is the axon, as we have learnt earlier. This is the axon, and obviously the plasma membrane of the axon will be known as axolemma. This is the plasma membrane of the axon. Okay, let's move to the next part. Now we need to actually push this diagram a bit down because we have lack of space for the labeling part. So I'm just editing it. Let's select this. No, not this way. Okay, we need to do something else. Okay, small and branched dendrites. So these are the synaptic vesicles. Now, axon neurons is the most sensitive part of the neuron, and the cell body, inside the cell body there are the nasal granules. The nasal granules are composed of ribosomes, which are free ribosomes. So let's write that as well. Free ribosomes plus rough endoplasmic reticulum. See, these are the nasal granules. You can see the dotted structures. These are the nasal granules that are present within the cell body or the cytosol. Now, which is the myelin sheath? This is the myelin sheath. Check. These are the myelin sheaths. See, this is the myelin sheath. So the myelin sheath is the insulated part of the neuron that doesn't allow transmission of ions in and out of the neuron. And these are the Schwann cells. I'm showing you. We have a lack of space actually. These are the Schwann cells. And the node of Ranvier is the portion where there is no myelin sheath present. So this is this part, you can call it, I'm showing it with a different color because we have lack of space. So I'm showing it with blue color. See this one. This is called the node of Ranvier. Node of Ranvier is where the myelin sheath is absent. So the transmission of ions in and out of the neuron occurs through the node of Ranvier. So you can see the entire structure of the neuron. Now it's very clear. Now there are several questions that can arise out of this triangle structure. The first question is that how is the myelin sheath formed? How is the myelin sheath formed? The answer will be, it's made up mainly of lipid. In the PNS, which is the peripheral nervous system, it is made up of, and obviously there is the CNS as well, So these are the two parts of the nervous system, PNS and CNS. You can understand. We'll come to it later on. But at this moment, I want to explain how is the myelin sheath formed. That's why I'm showing you the PNS and the CNS. There are myelin sheath. In case of PNS, it is formed by the Schwann cells, and in the CNS, they are made by the oligodendrocytes. Myelin sheath. The myelin sheath is the insulating sheath. Insulating sheath doesn't allow ion transfer. Next part, the nasal granules, free ribosomes are present, and rough endoplasmic reticulum is also present. Dendrites, I've shown you the cellular processes, the synaptic vesicle, the synaptic knob. This is the synaptic knob, the synaptic vesicle, and the axon terminals. The axolemma is the plasma membrane of the axon. The axon hillock, this region, is the most sensitive part of the neuron. The cell body of the cyton, composed of nasal granules, free ribosomes and rough endoplasmic reticulum, dendrites, and then the node of Ranvier, which is these portions, basically the gap where myelin sheath isn't present, and the Schwann cells that produce the myelin sheath, and these are the nuclei of the Schwann cells, and this is the main nucleus of the cell body. So we have got the basic, the detailed structure of the neuron, and now it's time that we understand the types of neurons. This will be a short lecture. I was planning to tell you about the nervous system as well, but this is the first lecture, so we will keep it short and keep it up to the types of neurons only, that is in one page itself. So types of neurons. Something is not working here actually. Okay. So, okay, types of neurons. Types of neurons, you have the unipolar neuron, the bipolar neuron, the multipolar neuron, and the pseudo-unipolar neuron. So, there are four types of neurons. The unipolar one, what happened to the pen? The bipolar, multipolar, and the pseudo-unipolar neuron. So, the unipolar neuron actually looks like this: a cell body, the axon, and axon terminals. The nucleus inside the cell body. We'll just rectify the diagram. It doesn't look good. This is the cell body. This is the axon, and we have the axon terminals directly without the presence of dendrites. This is the unipolar neuron. Now the question is where it can be present. It is present in embryonic condition. It is present in the embryonic condition. Next, the bipolar neuron, where the cyton is present, dendrite is present, and axon is present, but one dendrite is present only. Check. Okay, I'm drawing two axon terminals to ensure clarity here. So this is the cell body. and here you have the dendrites. So the axon is present and the only one single dendrite is present. And example, this is present in the retina, that is in the eye, and the olfactory epithelium. Olfaction means smell. Olfaction means smell. Next, the multipolar neuron, which is the one I drew on the top in a detailed fashion. This is the multipolar neuron basically. This is the multipolar neuron. Yes. This is the multipolar neuron. We'll just rectify it a bit. This is the nucleus, and we have multiple dendrites here. More than two. These are the multipolar neurons. Now the pseudo-unipolar neuron is an exception here, which is initially looking like a unipolar neuron, but later on gets branched here. One portion becomes the synaptic end bulb, while the other portion, it turns around into the axon. The other portion turns into the dendrite, like this. So this is a special case, and multipolar neuron is present in the cerebrum. Example, and obviously it is present in most of the cells of the body. And pseudo unipolar neuron is present in the example, the dorsal root ganglion. The dorsal root ganglion. So I've told you about all the types of neurons as well. We will just reduce the size. So it looks better. Yes, it now looks better. So I've explained to you about the neuron, the structure of the neuron, some basic questions, and obviously the types of neurons. Now this was the first lecture, so I'm keeping it short. I was planning to give you the basic terminologies as well, but I felt that this is the first lecture for me as well, so I have to take some time for preparation and then understanding the topic and then obviously explaining to you. It's not easy. It's definitely not easy, because after understanding to deliver it, it is the most difficult part. So yes, I'll be coming up with more lectures, and please share and subscribe our channel. Gradually more and more informative videos will be coming up. So yeah, goodbye and take care. Remember this topic, I'll actually upload the link of the Telegram channel or the Telegram group in the description box. You can go below and understand. When you browse to the Telegram channel, you'll be able to understand how the things will go in the future, the next lectures coming up, and so on. They will be uploaded in the Telegram channel itself, and thereby in the group itself. So thank you everyone, and please leave a like and subscribe our channel. Thank you. All the very best.